Thank you. And we were sort of comparing notes at breakfast, and it sort of come from these opposite paths where George Church grew up in a very non-academic setting and managed to sneak his way into it and do incredibly well. And I was the opposite. I grew up in a very academic setting and did everything to run away from it. So here we are.
Yes.
And DARPA, the old DARPA, rescued my childhood. I lived in New Jersey, and it was the most boring place in the world. And then one day, DARPA wrote their first check. Herb York was the scientific director. He was Eisenhower's science advisor. And he could write up to $1 million. He didn't need any approval. So he wrote these guys a check for $999,750 to start this thing that became Project Orion, a space project, and they hired my dad. So we got to leave and come to California. So a huge thanks. And I first met you through my sister Esther, who one day Science magazine called me and they said, do you realize this guy's he's publishing half your genome in public? Did anybody ask you? Because we had all these ethical questions about once we could do sequencing, what happens to this information? And is it available to insurance companies to say that they won't insure me because I'm a risk? So anyway, my sister was one of your test subjects of the Personal Genome Project, where you sequenced 10 entire people and freely published it, but indeed, nobody asked me. And, and I think they should have. So, yeah. But you're forgiven.
Esther was supposed to ask you.
Yes. If she had asked, I would have said yes. So.
Good. Okay.
So tell me, there was so much rich material this morning, but I think you're thinking bigger yet, which is, so the first question is why, you want to engineer entire genomes, not just doing this sort of editing. Right. And why do you think that's the path forward?
Right. Well, I think, we very much are in the groove of the DARPA, but maybe a little more so, and things like reversibility and safety and frugality don't seem like a path to a lot of Nature and Science papers, but it definitely is one of the things that motivates us. And the reason, you need to have a reason for some things. Pure science, pure engineering can be one of them, or two of them, but for whole genomes, we felt that we didn't want to make a copy of something, we want to make something new. The sort of new things that we could make would be something that was resistant to all viruses, not just the one-by-one cases that Dan was talking about, but and how would we do that? And then if we did that, we'd have to have some safety mechanisms. So it would be, and we need those anyway because we don't want drug resistance and pesticide resistance and so forth to get out into the world. So we need genetic and metabolic isolation, and that leads us to new amino acids, and they all get solved by potentially the same problem, same solution, which is recoding the genome, changing the genetic code, going from DNA to RNA to protein, so that you use the synonyms, and now incoming viruses or exchanging of genetic material becomes impossible. So I think those are some of the reasons that I came up for doing things on a genomic scale. But there are other reasons that we could go into, or how to do things on an ecological scale. Yeah.
And so that leads to the next question: is if that wasn't enough, that you just want to do entire genomes, as I understand it, and I've read a bunch of your papers on the trip here, you're not stopping at the organism. You want to engineer. Why not engineer the entire ecosystem to actually, with these gene drives, to actually go in and not stop. The way evolution works, in a simplistic sense, that it's driven by the organism, that if the organism survives, then the gene survives, and why not go beyond that and introduce genes into the environment that do something that maybe is not in favor of the individual organism, but is favorable to us, and that sort of changes the entire ballgame. Maybe you could explain that.
And to some extent, the same. If you're going to deal with ecosystems at all, you're going to change them. Even if your goal is just to manufacture something, you're going to change the ecosystem. We know that. And just like the previous topic about why whole genomes, the answer is safety. And that has to be at the forefront. And I agree with what Dan said. We need to have it as low impact and as reversible as possible. If you make it too pulled back, then you end up with a very expensive process that doesn't succeed. Dan mentioned that you know, a very small delivery, you know, inadvertent delivery of mosquitoes and/or malaria was enough to contaminate the Hawaiian Islands. So, likewise, a huge reduction is the same as leaving behind a small delivery, which then spreads again. So, how do you deliver something that will make the population resistant in a way that's cost-effective, reversible, and will essentially spread itself? And that's the gene drives. That was suggested for mosquitoes in 2003 by Austin Burt. I was introduced to it in my thesis in the 80s by Bernard Dujon in yeast, the basic idea. But the idea is if you have a package that is not in the best interest of the mosquito, you know, like malaria resistance, may be in the best interest of the birds or the humans, but not necessarily the mosquito, then you need to give it some Darwinian push. And that's this kind of this selfish DNA component, but with a package that's very far from selfish.
Yeah. So is that, and how has that worked out?
Right. So, I mean, so from Austin Burt's suggestion in 2003 until last year, there was, you know, progress, but slow. And then we had a revolution, and I'm sure many people know about, that we participated in, called CRISPR, which is a way of engineering a whole variety of organisms. It basically works in every organism that has been tried in, where you can get delivery. And initially seems like a good way of doing research or gene therapy, but it can now be applied to gene drives. And as soon as we realized it could be used for gene drives, we realized just how easy it was, and how, in principle, anybody with very elementary skills could make a gene drive. In fact, we even came up with a way that they could make them accidentally, just in the process. And then if they make it in a, you know, a mobile organism, mobile in any sense, flushing it down the drain or whatever, it could spread exponentially through the population. So our first papers on it were sort of detailed ways that it could go wrong, or you could reverse it, or ways of maintaining the safety. No experiments. So unlike some of the flu virus experiments, where the first the public heard of it was a fait accompli. And then now we published a paper. We did it in yeast in a very special way that made it so that it couldn't escape, even if it breached the physical barriers. And the yeast experiment was extremely rewarding in the sense that it spread nearly 100%, almost undetectable non-spread, and in a variety of yeasts throughout the phylogenetic tree.
Yeah. So when if all the stuff I read, the one that fascinated me most is trying to analyze the longevity of the bowhead whale. Here you have an animal a mammal similar to us, that appears to live upwards of 200 years. And you're looking at that, and the interesting question is, well, how do you do that so you can't wait 200 years to see if you're right? So how would you work on that problem when we're not going to be around in 200 years to congratulate you on being right or wrong?
Yeah, my department chairman wasn't too happy about having a bowhead whale facility either. But I think that longevity is a kind of a crazy goal scientifically, in that because even in short-lived organisms like worms and mice, you're talking about a payoff or an assay where just one turn of the design, build, test, learn cycle is longer than a graduate PhD. So what you want instead is not longevity, but aging reversal, where you're looking for signs of change that are nearly instantaneous, meaning weeks rather than years or decades. And this is perfectly reasonable. I mean, you can do it in model organisms. My close colleague collaborator, Amy Wagers, did what's called heterochronic parabiosis, where you suture together a young and an old mouse, their whole circulatory system, and you can find factors that contribute to the rejuvenation of multiple organ systems in the older mouse. And some of these have been followed up on in great detail, like GDF11 is an example of a protein that can circulate and has profound multiple consequences. But I think that's the way to approach it. Now, we not only worked on the bowhead whale, which lives 200 years, but the naked mole rat, which is dramatically different from other rodents. And I think as much as I've championed personalized medicine, I will be the heretic, contrarian to myself in saying that the thing that's wrong with humans, for the most part, is that we're human, if there's anything wrong with us. That is to say, the reason that we can't plow through a meter of ice the way the bowhead whale does from under it, or live so long, and the reason we're not normally adults the size of a mouse is because of human. It's not interpersonal variation in our genome. It is fundamental, and there are ways that we can change that.
Right, and what really makes us human, or above what we like to think makes us human, is our intelligence. And I understand you also want to mess with that. And now that
let's be reasonable.
Artificial intelligence, is getting a lot of attention lately.
Yeah.
But as I understand, your view is more that more immediate and more realistic is just slightly shifting our own intelligence, and what you told me was that don't bet against it, that this could be possible.
Yeah, let's lower expectations a little bit and broaden the scope. I mean, we heard earlier about machine-human interfaces and how great it is that we can now get a microscope the size of the end of our pinky, and optics is a great revolution. But the fact is that both electronics and optics are pathetic compared to biology in certain ways. In particular, the amount of code, the amount of digital information that you can pack into a biological polymer is on the order of 1 million times denser than any electronic components that we have. And the energy that is consumed in reading and writing to such biological systems is also about 1 million times better. And the cost curve, or the performance curve for biologicals, biology as technology, as Alicia will tell us, is not any slower than electronics, and some would argue is a bit faster, in that, when we started working on next-gen sequencing, the cost curve for electronics, which was a big part of next-gen sequencing, predicted 60 years, 6 decades, and instead it took about 6 or 7 years, okay? So we're ahead of Moore's law, and we are going faster. I will submit for consideration. I wouldn't call this improving our intelligence necessarily, although I do feel quite a bit more intelligent when I'm connected to Google than when I'm not. But I think that we have serious problems to deal with that are partly listed before that require biological interfaces with information. That information may or may not be in a digital electronic computer, but it's information.
So And you said in 2003, which intrigued me, where this has gone, it just seemed like a very bold claim. We suggest that the complete determination of a biological system is a concrete, achievable goal. And do you still feel that way?
Yeah, it's a little hard to provide context on that, but well, first of all, yes, I think it is. The simple answer is yes. And by complete, we're talking about synthetic biology provides us with some way of saying that we can reconstruct a biological system. That doesn't mean it's full understanding. It means we can reconstruct it. We could also say that we don't necessarily need to have full understanding in order to do amazing things. I would submit that we do amazing things with vaccines since the 1700s without full understanding of B cells and T cells and cytokines until recently. So what I think we can get atomic level information at this point. We have a new method called fluorescent in situ sequencing, which we did not exactly have in 2003. We had the idea in 1999, so that was in the back of our mind. But it allows you to essentially get the name of many of, if not all of the molecules and their three-dimensional coordinates. So I think we're on track for that kind of analysis. And synthetic biology will give us some feedback as to actually whether we can do the design, build, test, learn cycle on that complete analysis.
Right, which is one of the things you're working on is
right.
Make faster models.
To really know that you understand all the parts, you can make something that's radically different. Ironically, you know, if you make a mirror image cell, then you really haven't made a copy. You've made something with fundamentally new properties, but you've shown sophistication in taking it apart and putting it back together.
And how many people at work in your group? I mean, to me, is an important question. Sort of how large, you know, to produce all this.
Yeah. So our group is not a production group in the sense there are much bigger groups like BGI, which is a California, Shenzhen, China collaborative. But you could consider that's part of my group, right? Because I was one of the instigators of both companies before they merged. My formal group that I'm responsible for is 102 people in it, sort of equal graduate students, postdocs, and engineering staff. But I think you know, to innovate, you don't necessarily have to have the production completely integrated, although it helps. But you do have to integrate a large number of different disciplines, and so that's why you end up with that kind of force.
And how do you decide to divide your time between, which I find impossible. I can't, if I'm working on several things, I waste half my time trying to divide my time. And you seem to have some sort of gift for that.
Gift or curse. So, you know, I think there's only one project, so I don't have to divide my time. It's all one thing as far as I can see.
But you don't walk in the morning and there's several different people demanding you.
We take turns.
Yeah.
And we have you know, 6 speakers a week in lab meeting, and you know, I meet with companies when I'm in town, like today, and you know, it takes care of itself.
Yeah.
You know.